Welcome to the plant cell. We are about to embark on a journey throughout the cell. This is an example of what a plant cell looks like. On the outside of the cell is the cell wall. The cell wall provides rigid support to the cell and is made of cellulose, and is only in plant cells. As we work our way into the cell, we find the cell membrane. The cell membrane forms the boundary of the cell and acts as a selective barrier, allowing certain materials to pass but not others. The cell membrane is made up of lipids and proteins. Proteins assist in both passive and active transport. They occur throughout the cell membrane. Active transport is when a substance goes from a low to high gradient using ATP. Passive transport is when a substance goes from a high gradient to a low gradient without using any energy. The three types of passive transport are osmosis, diffusion, and facilitated diffusion. Next, there is the vacuole, which in a plant cell is very large and helps with structure. It is a membrane-bound sac that stores materials. Then we have the vesicle, which is much smaller than the vacuole, and there is two different kinds, the transport and the secretory vesicles. The transport vesicle carries what the endoplasmic reticulum produces to the Golgi. The secretory vesicle carries Golgi-modified products to the cell membrane, then it fuses with the cell membrane, releasing the contents to outside the cell. In the middle of the cell, there is the nucleus. The nucleus contains most of the genes that control the eukaryotic cell. Generally, it is the most conspicuous organelle in a eukaryotic cell, and it contains the nucleolus and chromatin. The nucleolus is found inside the nucleus and is where the components of the ribosomes are synthesized and assembled. Surrounding both the nucleus and nucleolus is the nuclear envelope. It is a double membrane that forms the boundary between the nuclear contents and the cytoplasm, and has pores to selectively let things go in and out. Attached to the nucleus, we have the rough ER and smooth ER. The smooth ER synthesizes lipids, including phospholipids and steroids, metabolizes carbohydrates, and detoxifies drugs and poisons, and even stores calcium ions. The smooth ER lacks ribosomes, while the rough ER has attached ribosomes. The rough ER channels proteins to transport vesicles, attaches carbohydrates to some proteins, and is involved in membrane production through the production of vesicles. The rough ER has ribosomes. Ribosomes are the site of protein synthesis and produce proteins for use either in or outside of the cell. Next, we have the chromatin. Chromatin consists of DNA and protein, and condenses to form chromosomes, and is inside the nucleus. Then we have the Golgi apparatus. It consists of flattened membranous sacs, receives transport vesicles from the ER, modifies ER products, and produces certain molecules and produces secretory vesicles. To the left we have an actual Golgi apparatus under a TEM microscope. The mitochondria is next. It is the site of cellular respiration and produces ATP from sugars, fats, and other fuels. Then there is the chloroplast. It's the site of photosynthesis and produces food using light energy, CO2 and H2O. Chloroplast is the reason for the green color. The region between the nucleus and the cell membrane is the cytoplasm, and it consists of cytosol. Lastly, we have the cytoskeleton. It is the backbone of the plant and is a network of fibers extending throughout the cytoplasm and composed of microtubules, microfilaments, and intermediate filaments. The differences between animal and plant cell are that plant cells have cell walls, which animal cells do not. Plant cells contain chloroplasts, while animal cells do not need chloroplasts and do not have them. But animal cells do have lysosomes and centrioles. But obviously, as you just learned, plant cells do not have either of those organelles. Finally, we have the cilia and flagella. The cilia comes in quantities of 100s and is on the outside surface of the cell and is small hair-like projections, while flagella comes in quantities of 1 to 2 at the end of the cell and is long whip-like projections like a snake.